Membrane receptors. After neuron transmitters are released into the synaptic cleft, they diffuse until they reach the postsynaptic membrane and binds with either a metabotropic or inotropic receptor. As the process of membrane receptor and signal transduction starts, a ligand, which is a chemical that cannot make its way through the phospholipid bilayer, binds to the metabotropic receptor that is located in the outside of the cell membrane and docks onto the heterotrimeric G protein, which is a membrane protein that is in the inactive state of bind guanosine diphosphate, GDP. There are three subunits attached to the G protein, which are the alpha, beta, and gamma subunits, and as the ligand attaches to the G protein, which is the receptor, it releases the alpha subunit and it moves towards the close protein known as the adenylyl cyclase. It bonds with an extracellular hormone which causes guanosine triphosphate, GTP, to replace guanosine diphosphate, GDP, on the alpha subunit. But before the alpha subunit gets to the protein, it is an inactivated enzyme, means it is not ready to work. As the alpha subunit is with adenylyl cyclase, it drops two phosphates from ATP, which is the energy, and converts it into cyclic AMP, also known as CAMP. Cyclic AMP are the messengers and they are going to spread throughout the cell. They are to target protein kinase, which is made up of different cells and subunits. Protein kinase A has two catalytic subunits and two regulatory subunits. Protein kinase A is going to take ATP and give out AMP, and it is going to take the phosphates and phosphorylate different proteins. Cyclic AMP are to attract to the regulatory subunit, causing the two catalytic subunit to release, which is called cascade. Cascade, as you can see, it changes from yellow to orange. From there on, the catalytic portions of the protein kinase are to become phosphorylated and pick up energy from ATP, act on enzyme within the cell, and drop off phosphate to phosphorylate. The activated pKa catalytic subunits enter the nucleus, where they phosphorylate Krebs, which is cyclic AMP, response element, and binding protein. And after all of this is done, the alpha subunit goes back to the G protein and everything goes back to where it belongs and the whole system shuts down, waiting for another ligand to enter.